Welcome to the Human Genome Project HGP, Genomics, Sequence Databases, and the BLAST Tool Introduction Lecture. The HGP aim was to sequence the entire human genome and provide the data free to the world. This was a global collaboration, the largest biological research project ever undertaken, involving thousands of staff over 13 years of work before a rough draft was published in 2003. It provided information on approximately 3 trillion base pairs and sequences of 30,000 genes. Crucially, the data is provided as free and open access through online public databases. The applications of the HGP are vast, particularly in molecular medicine, where it is leading to a better understanding of the fundamental causes of diseases, rapid diagnostic tests, and the ability to predict the likelihood of certain diseases. It also has applications in waste control and environmental cleanup by studying microbes with unique enzymes, energy sources by exploring methane production, and risk assessment by understanding genetic susceptibility to environmental toxins. Genomics is the study of whole genomes of organisms. It incorporates elements from genetics and is a computer-aided study of the structure and function of the entire genome of an organism. It deals with mapping and sequencing of genes on the chromosomes. There are different types of genomics. Structural genomics, the initial sequencing phase to determine the structure of every protein coded by the genome. Functional genomics, studying how genes contribute to biological processes, and comparative genomics, comparing whole genomes from different organisms. Genomic techniques have become indispensable in plant breeding and genetics. The era of genomics has led to the generation of enormous amounts of raw sequence data, necessitating the development of genome databases/biological databases. These are large, organized bodies of persistent data, usually associated with computerized software designed to update, query, and retrieve components of the data. The chief objective is to organize data in structured records to enable easy retrieval of information. These databases help researchers study available data, understand biological phenomena, act as a storage of information, and reduce data redundancy. There are two main types of databases: primary databases and secondary databases. Primary databases, also called archival databases, archive experimentally derived data submitted by scientists, such as genome sequences. Examples include GenBank, Nucleic Acid Database, PDB, Protein Structure Database, and UniProt-KB, Protein Sequence Database. Secondary databases comprise data derived from the analysis of primary data. They are highly curated and often use computational algorithms and manual analysis to derive new knowledge. Examples include Interpro, protein families, motifs, and domains, FAM, and Prozit. Searching and querying these databases for similar sequences is a fundamental task in bioinformatics. Given the size of these databases, fast search methods are essential. There are different search tools types, including similarity search tools like Smith-Waterman and heuristic search tools like FASTA and BLAST. BLAST stands for Basic Local Alignment Search Tool. It is a local alignment algorithm-based tool used for aligning multiple sequences and finding similarity or dissimilarity among various species. BLAST is a heuristic method, meaning it's faster and more efficient, but potentially less sensitive than some other algorithms. It finds regions of similarity between sequences by comparing nucleotide or protein sequences and calculating statistical significance. The process involves splitting the query sequence into overlapping words, finding similar neighborhood words, looking them up in a hash table to find their locations in the database. These are called seeds, and then extending these seeds until the alignment score drops below a certain threshold. To use BLAST, you typically enter a query sequence, select a job title and the database to search, choose a BLAST algorithm, adjust parameters if needed, and then start the search. BLAST returns the output as hit tables arranged by decreasing order of matched accession number, along with information like query coverage, sequence identity, score, and an e-value. The e-value, expectation value, represents the number of different alignments with scores equivalent to or better than the observed score that are expected to occur by chance in a database search. The lower the e-value, the more significant the score and the alignment. There are different BLAST program types depending on whether you are comparing nucleotide to nucleotide, nucleotide BLAST or BLASTn, protein to protein, protein BLAST or BLAST, translated nucleotide to protein, BLASTs, protein to translated nucleotide, BLASTn, or translated nucleotide to translated nucleotide, BLASTs. Thank you.